Welcome to this our Friday session on data analysis. One of our speakers was getting a little bit late here, so we're a little bit late getting started. Apologize for that. We're going to get started right now. It's a relatively short session. It's only an hour long, and then we'll have our three presenters here. One's just arriving, and then we'll do some Q&A following their presentations. Our first presenter is going to be Michele Reilly, who's the president, CEO, and founder of Turing Incorporated out of New York City. She applies math algorithms to challenging problems in life science and medicine. She began a current career working for Victor Niederhofer as an algorithmic trader, where she developed short-term strategies in the futures market. She has worked on modern methods and geometric flows and applied them to machine learning. And our speakers here. You want to, hold on, you want to give your PowerPoint over to the person in the middle of the room there? Sorry. You want to go second then?
I can. He's here, so I feel he may
We'll have James Watt go second. He's the general manager and chief scientist at Eagle Medical Services in San Antonio. James has more than 20 years' experience in life science research with a focus on infectious diseases and genomics. His current focus involves support in global public health programs for the CDC, along with helping them to expand EMS, Eagle Medical, work in stem cells and regenerative medicine. While at the University of Alabama, where he spent some time, he used genetic approaches to the discovery and development of vaccines capable of eliciting immunity to pathogens along with their delivery systems. And our last speaker today will be Michael Vassar, who is president and founder of MetaMed out of New York City. He's worked in molecular biology, genetics, and nanoscale physics, and has founded multiple startups and was executive director of the Singularity Institute, working with eminent scientists in all major fields. He founded MetaMed, recognizing that most revolutionary developments in life science never reach the public. We'll start off with Michele Reilly. Please welcome Michele Reilly.
Thanks everyone for coming. So I'm going to be speaking about data analysis needs in regenerative medicine and what we see as sort of a new computational paradigm for the life sciences today. So I guess this just forward, back, great. All right, so what are we doing? Here we are in the middle. What we have done is taken MapReduce, so basically an architecture, a distributed computing architecture that was created by Google. Jeff Dean in particular, who's sort of a demigod of the Google world here. So this paper is how to scale out clusters. So what we do is we provide cloud computing and data analytics for life scientists and other researchers. So you see here on the right is a optical scanning microscope, and one of the projects I was going to talk to you about today is a project with a company called Nanotronics. And what they're doing is basically they have hundreds of terabytes of data that they've generated from the Stephen Badylak laboratory sitting on their hard drives. And what we're doing is hosting that for them and looking at seeing what we can do to do what I'm calling empirical machine search. So how can we extract, so today, machine learning is very highly dependent on the prior assumptions of the scientist. And we're interested in how can we extract, so machine learning is about classification, categorization. Most of you are probably familiar with this already, but what we're in particular focused on is the idea of unsupervised learning. And so we'd like to sort of do a meta study whereby we're looking at inductive methods of hypothesis. So how do you take minimal assumptions of the scientist and try to basically understand what hypothesis generation, iterate on hypotheses. So you're looking at a vector space, a very large vector space, a heuristic search over a huge vector space of possible hypotheses. So that's when people talk about, you know, what is big data, to me it means this sort of a way of having the least assumptions as possible and just looking at this in an empirical way. There have been some pretty exciting results in unsupervised machine learning coming out of Google, especially with their AI research group, in NLP and also in deep learning. So I'm not sure if you guys remember, but in 2012 there was a paper about cats. So they took 10 million YouTube videos and They took 10 million YouTube videos and studied the bitmaps, and this was done in an unsupervised learning way where you're just sort of looking at these blobs of pixels, and they were able to identify, get some kind of accuracy rate for identifying these pictures of cats. So they're exciting breakthroughs in machine vision. We'd like to apply it to cell biology and see what can be done there. So traditional large-scale computation has been very processor bound. So I want to sort of explain the motivation for cloud computing or big data and how it could help life science. It's been very processor bound, so you send out your data, so you have to factor in the time that you send it to the processor and the time that it comes back. So there's like real high turnaround time, and so lots of complex processing, and we're only able to do it on very small amounts of data. Okay. If you've traveled in an airport, you've seen the Microsoft signs, welcome to the cloud, or what is big data. It's impossible to escape these things, but they're just sort of these very ambiguous signs. What does it mean? So basically, what you have is big data really means scalability. So when you need you need to scale your processes, you need to add nodes, you can add nodes more easily because of this idea of redundancy. So the architecture that we're using is redundant. The scientist can choose the level of redundancy and work with us exactly on the size of their data sets, what their exact needs are, and how to accommodate basically their image depth, their storage and compression. So we work with scientists on how exactly to get this done. This is what I consider to be sort of the paradigm for what is necessary to do distributed computing. So as I mentioned, we have data, we have a case study where we have data from the Stephen Badylak laboratory, where this extracellularized matrix tissue. And the question that we're trying to ask is, can we determine predictive features of this tissue? Is there anything that we can find out from this empirically to try to figure out whether the tissue will work or not work? So, as I said, we're using imaging, we're using microscopes from Nanotronics, looking at these data sets and basically partnering with multiple companies to try to use this in multiple cases. So we have a researcher who won an international pattern recognition contest for detecting mitosis cells, and you guys know more about this than I do, but mitosis cells don't look apparently very different from non-mitotic cells, and so we're sort of using this unstructured learning, deep learning methodology to try to work in other areas, and especially in regenerative medicine. This is a picture of the matrix. Here I'm listing what the McGowan Institute for Regenerative Medicine actually does. So they're working on whole organ engineering, cell matrix interactions and cell signaling, biomaterials, bioengineering as it relates to regenerative medicine. So some of the questions that we address for the scientist are the formatting and compression of the data. So we provide a data science strategy for scientists. Okay, so basically, the point that I wanted to make is that we have significant improvements in GPUs, the size and the speed of GPUs, and so we're able to leverage that now with our research and basically the amount of data that's available today. So, thank you.
It's good to be here. I guess you're pulling up my slides, but while you're doing that, I'd just like to thank the moderator, the speakers. I was able to actually ran into other speakers on the panel yesterday, and they're down from New York, and we had lunch, and we were looking for somewhere to eat, and we just stumbled into it was a Brazilian restaurant nearby, and so I was quite surprised. Y'all made me look like a vegan. I live in Texas here. That was great. But I'd like to thank a few people, the organizers of the meeting, Bernie Siegel. I'd like to thank Biomed San Antonio, President of Biomed San Antonio, Ann Stevens. Also the opportunity to speak here at this, and also the CEO, my boss, my two bosses there here to see me today from the Eagle office. But my talk today is on the data analysis needs in regenerative medicine. And so what I'm going to do is to kind of give you just a background and an overview of why I'm here today and how that is going to relate to stem cells. So let me try this. Okay, so in 2001, as a postdoc working at the University of Alabama, Birmingham, the pneumococcal genome had just been completed. Streptococcus pneumonia, which is a bacterial pathogen. It's a single chromosome, circular, and so you can see it's about 2.2 million base pairs, which is relatively small, very small compared to the human genome, much larger than a viral genome. But as a bacterial geneticist, I was asking hypothesis-driven questions about strep pneumo and how it could cause disease, how it could move from a normal commensal or seemingly normal commensal in your upper respiratory tract to moving into your lung tissue, brain, middle ear, and causing invasive diseases. And to do that, we had questions that had to be answered using modern molecular techniques. And with the advent of the completion of the sequencing of the genome, we were able to build the first pneumococcal microarray and do expression profiling. This is, just real quick, one of my figures disappeared there, but that's okay. This is a protein. These are one-letter codes for a protein called neuraminidase, which is on the surface of pneumococci. And this is a line representing that protein, the amino terminal, the carboxyl terminal, approximately a little over 1,000 amino acids. And this is a catalytic enzyme that we were studying. And so in order to understand its function, we were able to, with the advent of the genome sequence, go in and make deletions in different areas of the gene, and therefore it would delete certain parts of the protein. And then we could utilize that protein in an assay to see if it had lost any activity, or moreover, we made in-frame deletion mutants to see if this would cause disease. The strep pneumo would continue to be virulent even with the loss of function of this. So this type of study was made possible through genomic sequencing, and the fidelity of that sequencing is very important for you to be able to target these things properly. Okay. So that, in a nutshell, was some of what I had been doing previously. I was doing genomics in bacterial systems. So as you know, most of what we know from genetics today is we learned early on in bacterial systems because it was very simple. Currently, moving on toward the stem cell portion of this, I'd like to say that right now, the company that I support at Eagle Medical Services, and together the Eagle Group, we support the Centers for Disease Control in Atlanta. We have several programs ongoing there in the Division of Global HIV/AIDS, Global Migration and Quarantine, Global Disease Detection and Emergency Response. And a lot of these are programs that are utilizing informatics, not so much bioinformatics, to do surveillance and track epidemiological or pathogenic outbreaks and do epidemiological studies. So a lot of this isn't genomics, but it's taken a turn recently. This is walking into the visitor center at the Centers for Disease Control in Atlanta. You can see that there's constantly a slideshow that's moving, showing the CDC's presence in public health globally. And one of the things that is more and more important, as we all know, is that we live in a very small world and that we have to take global approaches to anything that we do. That is, when we do surveillance, and we try to find any outbreaks that may come into this country. And that's where we have our Division of Global Migration and Quarantine is very important. And it not only does the surveillance, but it has quarantine stations at airports in the United States where flights come in from overseas. And we have people that are developing tools for public health officials so that they can track in real time the people that would travel to this country that may have an infectious disease, for example, tuberculosis or something like that. So it's a large data set such as TSA flight information combined with electronic disease notification, big data sets. So those are overlaid to give public health officials the ability to do this. So this is some of the work that we've been doing at the CDC, and this is just a graphic representation of some of the flights. What you see in light purple are flights that are less frequent, and then the kind of the hot blue is more frequent flights. And so there's a great number of flights coming that travel all over the world, hence we can have outbreaks like we see in West Africa. And so just wanted to point out that it's well known that, and to quote this Suri Mahapatra, technology and innovation are key to smart global health. In the future, the use of novel diagnostics and the application of mobile phones and handheld computers will transform the system to one where patients are informed and empowered. A physician and health systems are engaged in understanding broad-based interventions for a wide spectrum of diseases. And we see that now, and we have surveillance systems in place that have done pretty well, but we're constantly trying to improve what we have. So we were because of our unique skill sets and backgrounds, past performance at the Center for Disease Control, we were recently, a few months ago, approached by the CDC, Office of Infectious Disease, National Center for Immunization and Respiratory Diseases, to support a new initiative called the Advanced Molecular Detection Initiative, or AMD. And so that is a 5-year initiative that is designed to bring genomic analysis, high throughput genomic sequencing that is done with next generation sequencing tools, and to bring that from a research laboratory and push it out to the clinics so that people that are in public health clinics can harness the power of whole genomic sequencing. Whenever we do whole genomic sequencing, obviously, like Michele was saying, is that we wind up with large data sets. So genomic analysis is the mining of genomic data. This is big data, but specifically when we speak of bioinformatics, we're speaking of next generation sequencing data. So this is a figure that describes the cost per raw megabase of DNA sequences. This was, again, this is put out. This was a slide that I downloaded from the NIH website. And so you can see on the left-hand side, 2001, moving over to 2014, and the cost associated with doing sequencing or whole genomic sequencing has dropped dramatically. That's through our improved technologies, chemical reagents, instrumentation, competition in the marketplace. So therefore, not only is the cost going down, but I think in this next slide, so this next slide you can see it's a similar figure, but they have the red line represents the number of base pairs per day. So not only the cost has gone down, that the number of base pairs that can be sequenced in a short amount of time has really increased. So I'm sure everyone's seen in the news that people are moving toward having their genome sequenced for approximately $1,000, and that is going to be something that is available to them to use in a personalized medicine setting. And so that's made possible through the advent of these novel diagnostic technologies that can rapidly sequence whole genomes, and the fidelity is fairly high. So the advantage of the whole genome sequencing is the workflows and approaches can be applied to a wide range of organisms, and what I'm getting to next will be stem cells. This is a fundamental cornerstone of omic approaches. It's very objective in that the sequence-based methods avoid subjectivity of phenotypic or fragment-based approaches. So we're no longer having to look at the phenotype, but we know the genotype in toto. So, and it's something that is the future, and we're there now. Some of the limitations, and this is part of this talk, essentially data analysis needs in regenerative medicine is the needs are what are limiting us right now, some of the gaps, and that is this technology is rapidly evolving, and so there's not standardization is something that's happening, but we're not there yet, and so therefore that's one obstacle pushing out to the clinics or to other private companies that do the whole genome sequencing. Comprehensive databases are still being built, but the baseline data is not quite there. And many analysis require specialized bioinformatic infrastructure and staff. And these people are, we have a team of these people at the CDC. I can tell you they're great people. They're computer programmers, bioinformaticians, mathematicians, statisticians, and they don't have a certification for the particular work, so they kind of fly by the seat of their pants, although they're very talented people. So the initiatives for the advanced molecular detection, and then I'll lead right into the stem cell and finish this up, but the overall initiatives is to form a consortia, partnerships, and align our efforts in academic, federal and state, nonprofit, commercial organizations, and to begin pilot projects for outbreak detection and leverage existing laboratory-based surveillance systems. So all of this new advanced molecular detection technology really centers specifically on next-generation sequencing and the analysis of that data. So that is something that's not unique to the CDC or to pathogenic microorganisms. We use that in stem cell research. So as we've learned over the last few days here, that stem cells have the remarkable potential to develop really into any type of tissue. So when you take a stem cell and put it in the presence of other cells that have already developed into a particular type of cell, a muscle cell, brain cell, then the stem cell can take on that phenotype, and it also has the ability to replicate, and it's a kind of a new fresh cell that offers promise to regenerative medicine. So the characterization of the stem cell lines is where we currently have a need. That is a limiting step. So the early work that tried to establish stem cell lines looked at it from a phenotypic view. So we're looking at just biological markers, and so we're really kind of blindfolded and just looking at a few pinpoint areas of a cell to try to identify it. And when you're in the laboratory and you're growing stem cell lines and cultivating them, and you do this in a continuous culture environment, the stem cell lines evolve. And that's not necessarily a good thing. You want a clearly delineated, characterized cell line, and that's necessary for it to carry out the particular function that you want it to carry out. And then, so we were able to take a large step past looking at things phenotypically and improve the technology. So these are just a few things that I'd like to point out. There's been a lot of progress just in the last couple of years in developed genome-wide assays and bioinformatic methods for the analysis of stem cells. Some of the original sequencing was doing global transcriptional profiling, so it was looking at the total RNA and looking at the profiles as a global transcriptional profile, which was a good measure, a better measurement and characterization tool for the stem cell lines than just looking at a few phenotypic traits. With the advancement of sequencing technologies, we have the global analysis of the epigenome. So we were able to look at the genome sequence, or the transcriptional profile, non-coding RNAs at the molecular level. So this really is helping. And so one of the tests that has been developed is called the Pluri test. It's an algorithm that makes use of training sets containing large numbers of undifferentiated, differentiated, normal, abnormal human stem cell lines. So you can do a comparative approach with your cell line, and it helps you assess and do a quality control measurement of your cells. So this is really important. For example, if you wanted to grow a whole organ using stem cells, and so you have to start with a seed population of cells that you may receive from another laboratory, or you may have purchased that commercially. And so it is very important to take good quality control measurements at the beginning of any experiment or any kind of commercial development, because like I said, evolution happens, contamination happens. And so if you don't do this quality control measure, then what happens is you can get to the end of your experiment or the end of your trying to grow particular organs or tissues or things, and you find out that it's not what you wanted and it's not what the FDA had approved. So that's really important, and it's going to save a lot of money to do whole genomic sequencing or the profiling of the transcriptome from the beginning. So next generation sequencing is a strong validation tool down in the nucleotide level, and it has become a reliable routine tool. Biomanufacturers can hardly ignore it as a means of assuring that cell lines behave as intended for the entire life cycle of the product or process. So the pharmaceutical companies really, they are getting on board with this. But again, this is a gap in stem cell research or regenerative medicine right now. So just the enhanced cost, speed, and fidelity of whole genome sequencing is something that we're continually trying to improve on. That's one gap. These large data sets and the stem cell libraries represent another mountain, and we have people like our speakers here that develop those type of tools to help mine data sets and improve our ability to answer hypothesis-driven questions or understand if a cell line is what it's supposed to do. So, and then also more powerful software tools. That's part of mining the large data sets. But essentially, I just wanted to say that the big picture here is that bioinformatics is the fundamental. It's a cornerstone of all the life science disciplines now. So whether it's infectious diseases, regenerative medicine, cardiovascular disease, or anything that you pick in the life sciences, we use bioinformatics together with whole genome sequencing as one of our most powerful tools that's helping transform science so that we can actually cure diseases rather than just treat the symptoms. And so I just want to say thank you. I appreciate that. Okay.
So I'm going to try to offer an extremely general top-level overview of the idea of data science. And it took me a lot of work to decide, but I concluded that the essence of data science was the idea of science without storytelling. So we're humans. Storytelling is really basic to us. A lot of how we navigate the world is we build these sequences of symbols that summarize different things that we perceive. And there's another way that we navigate the world, which is just perceiving without building any particular sequence of symbols and automated patterns of perception and action. We can use dialogue and discourse, or we can just also use our brains in a native mode. And when computers were first invented, they were very, very low power compared to today. And they were still much, much faster at certain aspects of the sort of reasoning, computation, explicit manipulation of symbols than our minds are. But they weren't able to do basically perception at all. But as we've gotten computers that are increasingly powerful, and at this point our most powerful supercomputers are probably in a technical sense more powerful than our human brains, that's really new. That wasn't the case 5 years ago. As we've gotten to that point, and as we've gotten the ability to store enormously much more data than our brains can store, something more like perception begins to be on the table. And so data science involves using something like perception from computers rather than something like reasoning. And someday we hope to bring the two together and have real artificial intelligence. But it seems like you can do a lot with just perception. So the problem with words is that they hide ambiguities. Science developed out of law to a high extent, which is an adversarial debate process. You have different people telling different stories and trying to bring the table facts that are inconsistent with opposing stories, winning battles for their story by a process of elimination. In this process, credit and authority for an individual are tied into their impact on the final conclusion, which gives them reason to expect bias. But the hope is that these biases can cancel out as long as no one has authority that's higher than the authority of facts. The Royal Society in England at its time of inception had a slogan, "Take no one's word for it." And it was dedicated to doing a sort of science that anyone could replicate on their own at home in a week or less. You know, maybe the hardest stuff involved building pumps and vacuums, and that might have required that you be able to spend a few years of wages for a peasant to bring in skilled craftsmen to build such things. But they weren't talking about Large Hadron Colliders or pharmaceutical trials or other situations where one actually has to take someone's word for it because there's no way to realistically reproduce the data. Anyway, a lot of the way in which people fight legal battles or fight political battles involves controlling the language, controlling the way in which perceptions, data, get compressed into symbols and words and narratives. And because we can store so much data now, a solution appears to this problem, which is not compressing the data into narratives at all, but just leaving it to stand for itself. If you have a petabyte of data, which today costs about $50,000, you can store basically any experiment, basically a lifetime worth of experiences, basically anything that you're likely to ever handle without needing to compress it at all. And you can compress it in a lossless manner, which doesn't require that you throw away anything, even more cheaply. And in fact, the compression in a lossless manner turns out to be basically the same thing as doing analysis. To a really significant degree, doing analysis means finding out that this part of the data is the same as this part of the data, and that is in fact how you do compression. So who in the audience feels that, for the most part, the legal system does a good job with scientific questions, like medical malpractice? Any hands? All right, didn't think so. The paradigm that data science is more like is, I'm holding up this piece of paper, and you're pretty far away, but you can probably read at least some of the bigger letters on this piece of paper. But then you look at the smaller letters, like say this word, and you probably have to stare at it for a little while before you read it. There's no adversarial process, but there is a process of incremental data gathering going on in your brain as your brain takes different information in through your eyes and puts together its best guess. And this is basically, unlike an adversarial process, it's a process that assumes cooperation. There's no system in your brain that is trying to make you get the wrong answer or a politically convenient answer. It just takes time, and you're doing this computation and building a best guess. But your brain is only holding on to information for about a second. No matter how long you stare at my hand, you're never going to see my fingerprints because the brain just doesn't hold on to enough data from my hand at that distance to make out a fingerprint. But in principle, if you could store all of the data coming from my hands, and people do this today with modern computer systems, you could do just that. Math is nice and objective. It's not a narrative form. It's kind of where we got the idea of objectivity from. You had some people back around 2,600 years ago coming up with this idea of an analytic proof, something that they could be more sure of than they had thought they could be sure of anything. And to this day, there's something of a fundamental divide between having a concept of certain, which means, no, I really believe it, I can act on it, and having a concept of certain that really is like, no, this couldn't be wrong. It actually, in a deep sense that's very hard to communicate, couldn't be wrong, which math tends to confer. Now, if you take away the narratives from science, what are you left with is basically correlations among your data sets, degrees of freedom in those data sets, different options for how the data sets can be compressed into n dimensions, and those dimensions can be characterized as Gaussians or other statistical distributions. Each parameter, a data set could be seen as a bunch of parameters, so that when we say that a bicycle, for instance, has, say, several degrees of freedom, 6 degrees of freedom, 8 degrees of freedom, whatever, what we mean is that there's a natural way of partitioning the bicycle so that the rotation of the wheels needs to stay correlated. The front wheel and the back wheel rotate together, and the handlebars rotates independently of, free of that. And semi-free of both, there's the leaning of the bicycle one way or another. I guess, so that's only 3 degrees of freedom. Plus you might say the orientation of the bicycle in space, and that would give it 5, and that gives you a very limited data compression that is reversible. You can take visual information in from a bicycle, convert it into a set of parameters characterizing it statically, and then reconstruct the information that came into producing the story, the model of the bicycle, from the story without any losses. Now, one thing about a mathematical world is that there isn't any such thing as causality in math. 2 plus 2 is 4, but 2 plus 2 doesn't cause 4. One of the things that we find is that causality, well, correlation doesn't apply it. It's always part of a story. It's an extremely useful type of story. It helps us a lot to make sense of things. But a lot of what we're doing when we talk about causation is we're talking about time. And another way of saying that is we're talking about throwing away the past. If you actually have enough information capacity to hold all of the past and don't need to throw it away, you actually don't need causation for the most part in your models. And once again, this makes things much more objective, much less contestable. Anyway, nice thing about math is it's much more reliable than mathematicians. You can see that way of reasoning when you try to apply it with words being very sloppy. You have Descartes trying to sneak in the idea of a judging, dying, and rising God from the idea of irreducible ignorance and a ground of being, because the way the words, the symbols, the compressions are made equivocates between these sorts of things. This is a part of what we're trying to avoid here. We're trying to avoid that sort of equivocation, even in, that even very fine minds, maybe especially very fine minds, are particularly capable of. Stories are useful as a way of compressing information. We've been doing something like this for a long time. Here you have a Ptolemaic data set, and you have the Copernican hypothesis, which is a story about how that data set is explained as the planets going around the Earth. From the sort of timeless data science perspective, I would say that the Copernican data set is no more valid than the Ptolemaic data set. What you're just looking at, can you predict things? And the Ptolemaic data set actually was a lot more accurate. We still haven't gotten to the point of generalizing universal laws. Machine learning is not going to invent the idea of an inverse square law today or anytime soon without having the idea that there ought to be a reference point for an observer programmed in separately, and without having the idea that there ought to be certain sorts of regularity programmed in separately, parametric equations, whatever. But so the sort of machine learning conception of science I'm talking about, the big data set, is incomplete. Another thing that we don't actually get is the theory of evolution. In the extreme limit where you store all of the information, Evolution is just a bunch of specific things that happened, a bunch of instances of some organism reproducing, a bunch of instances of some organism dying. And even the instances of organisms reproducing and dying are just sets of chemical events. As you can see, there is value in being able to reason, in being able to make systematic sense of things. But a lot of what we're exploring here is the value in being able to perceive without needing to reason, which has been basically underexplored until now, because until now there wasn't a shared self that can perceive. And until now, my perceptions were mine and not transferable except through my telling stories. But now, my data sets are mine and you can have them too. And they play a role analogous to perceptions. And, so we're exploring what can we do with that. If you don't have infinite data, of course, you do have to throw away data. And it's really useful to be able to create theories of evolution. Anyway, and we're never actually going to have infinite data, so we do want to develop that ability eventually. But our ability to gather data is going away. I mean, the limits to it. Like, sensors are a lot cheaper than eyes now. We can manufacture, the eye doesn't actually have that much resolution. I suspect, I haven't done the calculation, but I suspect that the resolution of all of the world's digital cameras at this point in aggregate exceeds the resolution of all of the world's eyes. I mean, iPhones have about a 16 megapixel resolution. And while at the narrowest focus of your vision, you can resolve about 600 dots per inch cleanly and pattern detect a difference between 1200 and 2400 dots per inch. Outside of about a 2 or 3 degree arc, you can't. So that doesn't actually amount to that many megapixels. And iPhones cost a lot less than 9 months to produce. So a great example in the real world of big data, perhaps the example, is the NSA, which can store information about every phone call that happens and can store all of the things that people say and their vocal tonality. And while they don't have the ability to check all of the words and tones against all of the other words and tones in every conversation, they do have the ability to compare the local connectivity graphs between conversants. And so if you have a pattern, a hub and spoke model, where one hub that connects to 29 other hubs that each connect to, of those 19 and 41 and 37 other hubs, and those connect in certain ways, and if that chunk of the graph appears in multiple places in the record of all phone calls, they can say, hey, those are probably the same hubs calling from different phones. And this can all be done automatically. And if you can do that, if you can identify people in that way, you can also identify a data set that characterizes the properties of one type of stem cell or another in terms of the actual volume, in terms of its reflectivity and refractivity, in terms of its tactile properties. You can reduce all of those things to numbers. You can characterize a tissue culture. And then when another tissue culture has the same properties somewhere else, it can be discovered in an automated fashion that these two tissue cultures are identical, that this anomaly has recurred somewhere. You can much more naturally pull together information from different experiments. So my company is called MetaMed, and we do, by hand, in-depth meta-analysis of the medical literature, so that, like Cochrane, we help people to make sense of a bunch of conflicting sources. But if scientific practice was just to directly submit the data, and if algorithms like the ones that the NSA and Google use were used on that data, the meta-analysis would be automatic. There wouldn't be discrete lines between publications. There would just be a giant pool of data with naturally emergent parameters and patterns within those parameters, and naturally emergent probabilistic updates towards the identity of one cluster of parameters with one another, so that when you learned something from one experiment, this would tell you more about what would happen on other experiments probabilistically in a data point by data point manner. This is taken to the extreme. Taken to the extreme, this is an absolute change in how we do science. I'm going to give one more example, which Michele mentioned earlier, nanotronics, because I feel that this is an instance where by using a big data approach, you actually throw away what I learned as laws of physics growing up. Like when I went to high school and college, I thought that you would never be able to use light to see smaller things than the wavelength of light. That this was an ironclad law like relativity and quantum mechanics. And just like you'll never go faster than light, you're never going to see smaller than light with light. But it turns out that even light, even matter, even atoms and nanoscale structures are in a sense a narrative compression. And while you can't tell a story about a particle diffracting and bouncing and see it as a single data point, you can statistically characterize how particles bounce off something and use a data set, how they are refracted, how they're bent, statistically characterize a set of optical detection events and use that statistical characterization to predict future sets of optical detection events. And this has unlimited resolution. It's limited only by how much data you collect and analyze. So it's possible to use a light microscope and see things that are 10 or 20 nanometers across, see features that I had thought you would need electrons for forever. And why are you able to do that? Because matter is a story. Matter is a narrative compression, but matter is a narrative compression that is robust enough that we know how to generate it with a computer. And instead of feeding the scientist using a nanotronics microscope a big data set which they can't use, the computer tells a story, generates a virtual environment that makes sense to humans of that statistical prediction as if it was, you know, what we think of it as being, a cell or a bunch of cells or a bunch of viruses. And this was used the first way, the H1N1 virus was initially visualized using a nanotronics microscope. This has been around for a little while, but it's mostly used for error correction on microchips, which of course allows us to get ever more data storage ever faster. Anyway, just a reminder, a petabyte is a billion megabytes. A megabyte is kind of a moment, a second of experience, more or less. So a petabyte basically can store a lifetime of experience. It can also casually store all the music, all the movies, all the books, and all of the human genomes in a single, I mean, there are more than a billion people, and each genome is a billion bits, but they're very compressible. And in particular, if you know someone's parents, you can really compress them a lot because you only have the crossover events and the mutations to store. So it actually only ends up being a few kilobits. The specification of parents plus a few kilobits per genome. You can, it's actually possible to store in a refrigerator size device at your home, basically everything that you can imagine ever wanting to know. So anyway, as a sort of anthem for this, imagine there's no papers. It's easy if you try. No authors or discussion, just data piled high. Imagine those citations speaking for themselves. Well, we don't need to dream. We can just look. Talk to you later.
Okay, we have some time for questions here. If somebody wants to ask some questions, there's a microphone in the center aisle there. Just try to address it to whoever are the speakers that you want to address it to.
Hi, my name is Michele. I'm studying the regulatory part of the translation part of regenerative medicine. And one of the main issues on the field is that stem cell therapies introduce a kind of inherent uncertainty on how it will work in a patient that regulators and sponsors are struggling on how to show evidence of the clinical outcomes that those therapies will have. How do you think that data science tools can help to mitigate or help to perceive better these kinds of outcomes on clinics and help to bring faster this kind of new complex technologies that will be always uncertain in the human body in a way that is continuing to be safe and working, so showing efficacy?
So,
the clinical part of the question. How do you think that data science tools can help in the clinical approach?
I would think. I would think that from a clinical perspective, you know, you're obviously developing priors. You have some notion of what you know and what you don't know, and you're oftentimes in these large data projects, we're building what we call Bayesian networks. Are you familiar with it? Yeah. So I mean, there's really like a scale distinction between clinicians and getting large data sets, but I feel like the bridge between those two are these sorts of microscopes, so the what nanotronics is doing, for instance, and just amassing this huge amount of data for imaging purposes, and then you can build your BaseNet on top of you know your priors, insofar as you want to build in your priors, or you know just look at it from a frequentist perspective on just sort of like, you know, what do you see at this nanoscale?
So I think that there are two questions that data science is relevant to with respect to collectively perceiving the clinical outcomes. The first has to do with throwing away categories such as adipose stem cell and better characterizing the actual reagents, the actual materials that are used, having gene expression profiles, having epigenetic data, rather than having a brand name. One way of thinking about this is data science can be good for replacing brand names and stories about what was done with quantitative characterizations of what was done. And this is important because a lot of the time, whole fields of research get killed off by people doing things that aren't actually very related to those fields of research, but just sloppy, silly things that are given the same names. And that's the low-hanging fruit in the field, is moving towards a norm of characterizing reagents rather than naming them, characterizing physical activities rather than naming them. Like, characterizing surgery is going to be hard until we have fully automated surgery. But all of the things that are put in a body, we ought to be giving, you know, test data on the thing. Sensors are becoming cheap. We ought to be able to just show these are the cells that were put in the body. Farther down the line, we have to deal with the question of acknowledging outcomes. So the current scientific community is not just uncertain about what was done in a given experiment. It's prone to not acknowledge outcomes, even when the outcomes are flagrant and large. And that isn't a problem of statistical analysis. Fundamentally, it's a problem of trust and ego, and of the entanglement of prestige with accomplishment or with presentation of data. So a social norm that separates identity, ego, prestige from the presentation of data would generally allow large we outcomes to shine through, and we wouldn't have the ridiculous situation where, you know, we have someone showing the statistical effect size of synthesizing a replacement penis on a mouse or not in Atala's talk, and saying that there's many standard deviations difference between the amount of regrowth that happens when you synthesize the new organ or not. Well, yeah, that's a ridiculous thing to show statistically. What we're really doing is having a set of norms that are aimed at detecting subtle effects being misapplied for detecting large effects, when the question is not one of, can we really detect something, but do we really trust you? And, you know, if you show all the data, then you don't need any trust.
James, you have anything?
I don't.
Okay. Anybody have any other questions? Okay, I have one question I'll close it up with. Do researchers have the ability to fully manage or understand the data they generate, or do they need outside help in general? Anybody?
In general, even the best scientists are not going to be familiar with computer science and mathematics and signal theory, and it will be very helpful to build tools and cross-disciplinary collaborations to let them make better use of it, at least in the medium term. Hopefully in the long term, there was a time when even the best Greek philosophers like Aristotle weren't familiar with arithmetic, and it was specialized knowledge to be able to multiply together, two three-digit numbers, or what we would see as three-digit numbers in a world with a zero. We can hopefully eventually develop better ways of communicating these ideas and teaching them faster and as a more basic part of their scientific education.
I mean, I think you're seeing often surprising correlations in the data, and you can have, and the researcher sort of, the human element is essential in yay or naying it, and I don't know, being that clinician, or having that knowledge to say, okay, let's explore this, let's run another test. So it's, I think they're important.
James?
Yeah, I would say yes. The more diverse group of people that you can have interacting with the scientists always leads to a better grip on the work. Having people there to constantly consider other ideas and concepts certainly gives you a stronger scientific team. So.
Okay. All right, I want to thank our panelists and for being here, and for the audience as well. And thank you, and we'll end the session right now.